The long strand of DNA double helix are tightly rolled up around these proteins called histones. So they're well organized to 46 chromosomes. All of our genes are carried within them. What is a gene? Anybody? You guys are so shy, huh?
Contains information to make glucose. Is that the definition that you're looking for?
The gene?
Yeah.
Yeah, it's the DNA sequence that makes one protein.
Protein.
So if we look at all the DNA strands, we go, well, who makes the hemoglobin that carries the oxygen? So we got to find that thing. We found that one thing. The one strand of DNA that makes that protein, like the hemoglobin, is called a gene. And so we have many, many genes.
Is it a strand or is it an area on the strand?
It's an area on the strand. It's not a strand, it's an area on the strand, yes. Chromosomes carry the hereditary characteristics. One gene contains information for one protein. That's really most important for us. We have 46 chromosomes. We have 2 sets of 23, maternal and paternal. And another verb that we use for that, meaning we have one of each parent, we call that, we are a diploid, making us a diploid organism. Because we have 2, di means 2. If we only had one set, like other organisms do, they call those haploid organisms. Half, as in half. We have about 30,000 to 40,000 genes that is packed in the double helix that you can see, right here. That's made up the double helix. And these are these histone proteins that they're nicely coiled around and wrapped around. And in a normal state of the cell, they kind of look like a bowl of spaghetti, the way they're in there, wrapped around the histones, about 2 meter length. And so that's known as chromatin, the spaghetti is chromatin. And when you get to cell division, then we get them organized in this weird looking 2-legged structure that is known as the chromosome. And that's where we see all these, when they line them all out and say this is the human genome, that's how they show them in chromosome format. Because then they're organized, then we kind of understand what we're looking at. A centromere is going to be important in cell division, and that's the point where those 2 legs connect on the inside. And then what we need to do basically to make the protein now, we need to find that part of that chromatin where the protein's at, or that gene. We need to uncoil it. We need to, and then we need to make a copy of it. And the copy is going to be then called RNA. And that copy goes to the ribosome, and then we can make a protein. The double-stranded helix is interesting. Each strand is a specific sequence of 4 nucleotides called adenine, thymine, guanine, and cytosine. 3 in sequence, so this is the magic word, 3 in sequence code for one specific amino acid. So 3 goes 1. There are 20 different amino acids in the body that make up a large variety of proteins. What do we call the 3 nucleotides in sequence? You should know that. I underlined it. A codon. Good. Or sometimes they call it a triplet. Good. The triplet stands for the fact that we need 3 of those nucleotides to make up one amino, or bring in one amino acid into this chain of amino acids. And so when we look at that, we basically have in the gene pool, we have all these, that's the code. The genetic code is all of 4 letters in sequence. Just depends on what sequence and how. And you better not make a mistake, but it looks all like this. It's almost like a computer with binary, it's just quaternary or something. And then we have the ability now to figure out which nucleotides in sequence code for which protein. Which amino acid. And then we can make tables, we can say, well, if it's a a GAC is going to be aspartic acid. And we can figure out these tables, and then we can basically, go nicely about and manually figure out which amino acids is coding for what. We're not going to go through this process. I'm not going to make you memorize all these 20 amino acids. Thank you. Does that make somewhat sense? Good, because that's the foundation of what's coming next. Oh, look at that. What's a DNA copy called? RNA. So the only difference between DNA and RNA is the sugar. It's the way the sugar looks. And I don't have it here, I don't think. No, that's good enough. So, let's make a protein. Let's figure it out. These are the steps. First, you need to find the DNA sequence. You need to unbind that DNA, double helix, where the specific gene is. Then you need to make a copy. So first you need to unbind the DNA. Then you need to make a copy of that gene, and you make that from a DNA to an RNA molecule. And then that RNA is a copy, so it's okay if it's going to be destroyed. It's not the original. That RNA goes out of the nucleus, exits, and feeds into the ribosome. And then in the ribosome, you have, or you feed it in, and then there's a different molecule. We'll talk about that in a minute. It's actually a different RNA molecule that then reads on one side, it reads the codon, and on the other side, it knows what amino acids is specific for that codon. And then it brings that amino acid onto the chain where it's needed, when it's read at the right time, and attaches the amino acids to the previous amino acid. So that's how we're going to make that little chain. Yes, sir.
Does the ribosome have to be free-floating or the...
That's either or. The way of making the protein is the same. And here are the different RNAs. We have 3 different types. I'm still not quite sure why they call them all RNA, but this is one where I just like, okay, let's just study it. Okay? So just take this one out. It's not that complicated. The mRNA is the one we mostly talk about. That's the messenger RNA. That's mRNA. That's the copy of the DNA. That's the copy of the DNA. Then the ribosomal RNA is rRNA. And that's part of the ribosome. That's all I'm going to say about that. Part of the ribosome.
Is it for making more of just itself, maybe? Is it just for making more ribosomal?
No, it's just part of the ribosome itself. It doesn't make anything itself. It doesn't make more of itself. It's just a structural piece of the ribosome. But I'm not quite sure which structural piece it is. I can read all more on that, but I'm kind of going over one. And then the last ribosome we have is the transfer or the tRNA, and that's the one that reads the mRNA on one side, and so it has one side is a triplet or a codon, and the other side is an amino acid. And so it attaches on the bottom, it reads the codons, and on the top it drags in an amino acid. And then when they come in next to one another, the amino acids just end up being a bead chain that comes out at the end. And that amino acid chain is the fundamental structure of the protein. And so from there, everything comes from there. So 3 types of RNA. So the DNA copy, once it's done, which is the RNA, leaves the nucleus and feeds into the ribosome, which is the protein factory. Then the other molecule, which we talked about is the tRNA here. This is the other molecule. Collects the different amino acids and reads the codon sequence, and then has the ability to attach the one amino acid to the other in the proper sequence. And that from there we get a polypeptide chain. Remember, a polypeptide chain is basically an amino acid chain before it's a protein. It's just the smaller ones are polypeptide chains, and once they say they reach 50 amino acids, they call it a protein. What is the second stage of protein synthesis called? So we go through the protein synthesis, we got the copying stage and then the making protein stage. Translation. Good. So the same process that I described before, we just have two extra terms. So making a copy in the nucleus, making from an mRNA from a DNA, and then sending it through a port to the outside, that is known as transcription. It's like court transcriber. It's still English, but it's just typing it in a different format. So that's a transcription. The DNA and the mRNA is the same thing. It's just a different molecule holding the same information. It's still the same language. It's nucleotides. But then, when we're on the outside, and we get that mRNA strand going through the ribosome, and then bringing in an amino acid, now we're going from RNA to amino acid. Now we're going from English to Spanish or French or something. This is like nucleotides and then amino acids? That's like completely different. And so that's why they call that part of the process translation, translating into a different language. Does that make sense in that format? Good. And I guess that comes back over and over because I just had a bunch of this with the micro first couple chapters of the study, and it's pretty much the same stuff. And depending on where you go, if you go to micro, you just pick up more on it, and hopefully, because then you get some chunky genes, some other reverse transcriptase, and all kind of weird stuff that can happen. But fundamentally, this is how the body can make protein.
Maybe this is a stupid question. So when we eat protein, it's digested and metabolized down to just the very building blocks of amino acids, and everything is completely reconstructed again using. And of course, we have to make somehow the ones that.
In order to absorb and assimilate the protein, it's pretty much. I don't know if it's going to be the di-amino acid type state or whatever, but I think it's single amino acids that we get through the gut lining. I mean, glucose, I remember, it's single glucose that we have. Monosaccharides need to come through. And so we are not looking at sections and segments that we can then take fragments. We're looking at recreating and restructuring from the beginning. All right. Tired yet? No, I am. 18, we got a few more going. It is important for cells to divide, and make 2 out of 1. Before you can do that, the hereditary material, the DNA, has to be copied. Once that's done, the 2 DNA copies need to be separated in a way, that's where the central thing come back. They need to be separated in a way that each daughter cell ends up with a full set of DNA. All right, full set of DNA. What are the steps called to describe the delicate process of doing that? Prophase, metaphase, anaphase, and telophase. Good job. PMAT. I made it up, or somebody made it up in Cairo school, and then in the beginning when I did these tests, I took the test myself. You guys take it to know if I actually know the questions I'm asking. And it came like to one of those mitosis questions, and the thing comes to mind is PMAT. I was like, I've done this 15 years ago, I still know that. And then I could reconstruct what that meant in my head. And so the mnemonics, I realized that mnemonics are very powerful.